Thank you everyone for coming to my exit seminar. I'm very pleased to see you all. When I was finishing my master's degree, I started to think about using remote sensing and GIS skills that I learned to use in ecology. And I started to look for PhD opportunities, and I came across to the project that Kara announced. And it was about remote sensing of wildlife habitat. Basically, investigating animal and plant relationships using remote sensing. Then this reminded me Tobler's first law, which says everything is related to everything else, but close things are related more related than distant things. So then I thought that if there are animals in the landscape, there are some landscape attributes which attracted those animals. It wasn't that easy to find those relationships, believe me, it was quite difficult. And so my talk is about habitat highs and lows using multi-platform LiDAR to assess relationship between vegetation structure and landscape used by birds and reptiles. I will start my presentation by giving background information, then I'll introduce my thesis chapters, then methods that I developed and key findings, and I will give some recommendations for the future research. My study area is well known, Mulligan's Flat and Guru Nature Reserves. It's critically endangered ecosystem. There are a number of restoration activities have been conducted, including introduction of coarse-woody debris and exclusion of feral animals and some grazers and so on. And there are 96 1-hectare plots. These small rectangles are size of 200 by 50 meter. So there are 96 plots, and they are clustered based on 4 types of vegetation classes: high tree, high shrub, cover, high tree, low shrub, low tree, high shrub, and low tree and low shrub. Basically different types of vegetation, and they're clustered, every 4 plot clustered based on the same vegetation class. And we have 96 plots and 24 clusters scattered. So what is vegetation structure, first of all? It is this horizontal and vertical distribution of plants across the landscape. They are very important for different species. They provide food, shelter, and microclimate and nesting sites for different animals. And therefore, habitat assessment methods require accurate assessment of vegetation structure. However, collecting information on vegetation structure is very time-consuming, tedious, and resource-intensive. Imagine if you have some technology which gives you whole three-dimensional model of the landscape. First one is terrestrial laser scanner. We call it point cloud, or I call it shortly TLS, or I can call TLS LiDAR. LiDAR stands for light detection and ranging. It basically technology just sends laser pulse and creates lots of points which represent three-dimensional model of the landscape. First one is TLS, then the second one is ULS, UAV LiDAR, or we also call it ULS. So this is also giving very detailed information, however, not as detailed as TLS, as we can see. So there have been some few researches I saw during my study. So, however, all of them have been using airborne LiDAR technology, but airborne LiDAR gives you only a few point cloud, which means that they cannot detect below ground. And they only can give you, most of them can give you only information on the top of the canopy. So, however, on the other hand, we have lots of ground vegetation or ground layer attributes, and they are very useful for different animals and different species. Therefore, we decided to use these higher density point clouds to investigate plant and animal relationships. I have 4 thesis chapters. First one is comparison of TLS and ULS data for wildlife habitat assessments in temperate woodlands. I presented this work in IGAR's conference, and it was accepted for proceedings of the conference. And the second one is comparing TLS and ULS data for modeling avian species richness and abundance in a restored woodland. Then third chapter is multi-sensor approach for detecting coarse woody debris in a landscape with varied vegetation cover. Using LiDAR, then the fourth chapter is using LiDAR-derived landscape structural metrics to explain occurrence and abundance of reptiles in grassy woodland. I almost completed my fourth chapter, but there is still a bit of work should be done. Therefore, I won't be presenting this work today, but I still have lots of things to tell you with my 3 other chapters. So I will focus on only 3 chapters today. My first chapter was to compare the performance of TLS and ULS LiDAR in providing vegetation structural metrics to assess habitat quality. On your left-hand side, it's a drone LiDAR, it's a little animation or ULS. And on your right-hand side, it's a ground LiDAR. It's a small animation, how it works basically. And as you can see, TLS LiDAR is giving a lot more information, and especially in the ground layer. So ULS data collection took about a week to collect all these, to cover all these 96 plots. And TLS data collection took about a month to collect 7 scans in each 96 plots. So there were about 700 scans, which were terabytes of data. And we first conducted some pilot study to see how many stations do we need to collect in each 200 by 50 meter plots. So that we scanned like 4 stations, 5 stations, and 7 stations. And we saw that to be able to cover all these, to collect data, like appropriate data from these 96 1 hectare plots, we needed at least 7 scans. Therefore, we scanned 7 scans in each plot. After lots of data processing, like merging, removing noise, and etc. And this is the visual comparison of TLS and ULS data. First of all, the gray image is basically colored by height. So lighter gray shows the higher points, and lower points are black, basically. And you can see some occlusion or obscured spots in the TLS. It's caused because of TLS cannot see if there are some bush or more ground layer vegetation, TLS cannot see. Therefore, it has some obscured spots. In the middle image, it's a ULS data or UAV LiDAR data. As you can see, it's giving more coverage than the TLS data. And the bottom image is showing the difference between TLS and ULS. Blue to red areas showing basically the difference, which TLS couldn't capture, however, ULS could capture. Then we extracted 37 variables, which shows the vegetation structure. Basically, we first stratified the layers stratified the landscape into different layers. Ground layer is up to 1 meter, then mid-story 1 to 10 meter, then upper canopy above 10 meter. So we calculated vegetation volume and different height metrics like maximum height, mean height, or percentiles, and Shannon diversity indexes, which basically shows the diversity in the vegetation structure. Then we did some statistical analysis to compare the metrics derived from both sensor data. So key findings is vegetation height was generally higher in ULS than TLS, because since TLS is in the ground, if there are lots of trees, it cannot see on top of the tree. And also TLS could detected more structural information under the canopy, because again, it's on the ground. And again, canopy cover was also completely captured with TLS. Then also TLS captured more variation in the vegetation structure, especially in the ground and mid-story layers. So, and also TLS reported higher vegetation volume than ULS. Therefore, we expect that in our analysis, TLS could give like more accurate prediction power than the ULS. So my second chapter, aim of the second chapter is comparison of the performance of TLS and ULS LiDAR-derived landscape structural metrics for modeling bird diversity, functional traits, and species level bird abundance. Our first hypothesis, I'm using some trying to use some animations because I have two kids watching, and I don't want to hear like, "Daddy, what a boring presentation afterwards." So my first hypothesis is TLS data will perform better than the ULS modeling over bird abundance, species richness, and diversity. As we have seen, they give like higher values. TLS gave higher values in vegetation structure. Then our second hypothesis is bird species and gills that use lower layers would be modeled more accurately than with TLS, and birds that use higher canopy would be modeled with more accurately with ULS data. So therefore, for this, we needed to select models. We have 37 variables. We decided to remove highly correlated variables and keep only like more ecologically meaningful variables. We used Pearson correlation matrix, and select threshold, correlation threshold was 0.7. And we ended up getting 12 out of 37 variables as predictors in our model. Then one might say, so you have like 37 variables, and you are removing like most of them, and they might explain something in your data. Therefore, we tried PCA model as well, and principal component analysis is used to reduce dimensionality of large data set by preserving most of the variation in the data in the first 3 to 4 axes. So we calculated PCA from all LiDAR variables and got four PCA axes as predictors. Remember, we have 4 vegetation types: high tree, high shrub, high tree, low shrub, low tree, high shrub, and low tree and high shrub. This is the core of design of Mulligan's flat plots. So all of those 96 plots were based on these vegetation types. Therefore, we thought that if whichever the model can predict these vegetation classes accurately, more accurately, that model should be used for predicting birds as well. Therefore, we used multinomial model to classify vegetation classes from PCA and also 12 selected variables. As you can see, 12 selected variables gave more accuracy than the PCA variables. And also we tested bird prediction model using a PCA and 12 selected variables. Again, 12 selected variables gave more accuracy, therefore more prediction power, therefore we decided to use 12 selected variables. So from now on, I'm only talking about 12 variables. We used mixed effects model. 12 selected variables were fixed effects and 24 clusters were random effects. Response variables were bird survey data from 2017 to 2019. So this shows the comparison between TLS and ULS derived selected variables. As you can see, most of the variables are very close, the values are very close. However, if you look at the volume and lower layer vegetation complexity indexes, TLS is giving much higher value. So overall, we first modeled all of the birds that were observed between 3 years, and these basically symbols are showing different canopy structural metrics, and this orange line showing the significance level. So if the variable is above orange line, it means it's significant predictor. So as you can see, abundance didn't respond to any of the selected variables. However, species richness, bird diversity, and other diversity indexes responded to one or more variables, predictors. We also modeled the bird abundance within functional groups or guilds. So we grouped first birds based on the different habitat classes like grassland, woodland, and nesting, and also foraging guilds and some dispersion guilds. As you can see, all of the guilds responded to one or more structural variables. Then our hypothesis was, if you remember, was like birds that are associated with ground layer would be modeled more accurately with TLS than ULS. However, our model is showing that if you look at this explained variance, they are very comparable for ground nesting. However, for ground foraging, ULS is outperforming the TLS. And also, another hypothesis was like upper layer dependent birds would be modeled more accurately with ULS than TLS. Again, the result shows almost very similar results. We also modeled individual bird abundance of 41 species. If I would include 41 species, it would make this slide very busy. Therefore, I just selected mostly, like first row shows the birds that responded that give highest explained variance, basically. And the second one is least performed models. As you see, highest performed models gave lots of prediction power, and explained variance ranged between 70 to up to 80%, which is very promising. And those second row is showing least performed models. Explained variance was around 5 to 10%. So our first hypothesis wasn't supported because we have the similar results for TLS and ULS data. And our second hypothesis also wasn't supported because, again, guilds responded almost similarly. So what are the take-home message from this research? Habitat is species specific, which means you cannot model all the birds with one model. So models perform better for individual species or functional groups rather than overall bird abundance. Or if you include all the birds, it didn't explain anything. But if you go into guilds and species, then you had some explained variance. Overall, there wasn't big difference between TLS and ULS models. Therefore, ULS is faster and TLS LiDAR than the TLS LiDAR, and ULS may capture sufficient details of vegetation structure that's used by many of the bird species. So recommendations for the future work. I didn't use machine learning methods for this work, and because machine learning can include all the variables and give you the prediction power and give you it selects some important variables, and I didn't do that. That should be tested like random forest or neural network or support vector machine and so on. And also fusion of TLS and ULS might improve the prediction power. I didn't use that data either, but I'm using it for my fourth chapter, which I want to present in it today. So this work has proved the rich data set, provided the rich data set for future studies to which we can look at the finer scale relationships between individual birds and vegetation structure. My third chapter is to develop a model to identify and quantify coarse-woody debris from TLS and ULS, and also fusion of both data set for this research. I merged TLS and ULS. It was quite a bit of work to merge them. When we visited first this coarse-woody debris, it was like, when I saw them, lots of firewood, like, because my background wasn't really ecology, and it requires so much work, and there wasn't much work because people people are using this technology for only standing trees, and not many people have used this for laying trees. Therefore, I needed to develop the new method for myself because there wasn't much work on it. So as you can see, coarse woody debris can come clumped, dispersed, and combination of both types. So for this research, first I calculated digital surface model from points up to 1.3 meter, because from the field work I know that most of the height of most of the coarse woody debris is up to 1 meter. And I then calculated like different topographic variables, including like surface roughness and topographic position index and slope aspect and so on. They're just raster images. Then I trained the data by delineating small polygons of coarse woody debris and the grass and shrub in the digital surface model. Then I just ran random forest classification, like machine learning classification algorithm. It gave me some classification map, and we used some post-classification smoothing to remove the noise, and also exported coarse woody debris class candidate pixels to shape polygon and try to detect like elongated shape polygons. And we also calculated the volume of the coarse woody debris. This image shows the DSM derived from DSM. It's a shaded relief. You can see some coarse woody debris, and detected coarse woody debris looks like something like this. And you can see the difference between TLS, ULS, and fusion of both data set. The cyan color shows the coarse woody debris that I measured in the field. So they are the geolocation of each coarse woody debris. And detected coarse woody debris looks like this, like there some polygons. And we also, for ground truthing, we selected 18 sites, including different types of coarse woody debris and different types of vegetation cover, ground vegetation cover. And we measured locations, length, and diameter. And overall accuracy is, we calculated completeness and correctness accuracy. Completeness is giving like true positive divided by true positive plus false negative. True positive is correctly identified coarse woody debris. False negative is which were measured in the field but was not detected by the algorithm. And correctness is true positive divided by true positive plus false negative. False negative is falsely detected coarse woody debris, basically. If you look at this image, TLS is giving very varied accuracy. Basically, completeness and correctness should be higher. So this is the main point of the accuracy assessment. So accuracy by different ground vegetation cover, we identified. Again, completeness looks pretty much similar. However, if you look at correctness, high vegetation cover areas are decreasing the accuracy because algorithm couldn't really differentiate coarse woody debris from some tussock or for high density grasses. We also calculate volume of coarse woody debris. It's again, R-square is quite promising for especially ULS and the merged data. So key findings were overall developed method gave varying results from 30% up to 80%. And ULS sensor was better at performing coarse woody debris from ground vegetation, which provide better accuracy, correctness accuracy. So CVD detection accuracy highly influenced by ground cover vegetation, and this is reflected mainly in the ULS data. So for future work, different interpolation methods should be tested to increase the accuracy. And also LiDAR gives intensity information, which LiDAR pulse, like reflected pulse from different objects gives different power. So sensor records, and it might improve the modeling accuracy. It has been quite quite long journey, and lots of people helped to conduct this research. First of all, I thank Australian National University and CSIRO for funding this research, and I thank Kara for her support and encouragement throughout the years. She's just super supervisor, very supportive. And I had outstanding supervisor, Sean and Justin, Tommaso and Justin. They are very supportive. I learned lots of skills, lots of new things from them. And I thank Adrian, also Michael and William Foley, Professor William Foley for the valuable advice. And I had amazing collaborators, Martha and Tim. I say big thanks to them. And my supporters, Pat, Christian, and Kos. Thank you very much, all of you. And big thanks to Scott, Michael, Jack, Wes, Audra, and Ben for their support throughout the years. And I had amazing volunteers who helped me to collect huge amount of LiDAR data. Terrestrial LiDAR data collection took a lot. So I say big thanks to them as well. It was amazing work, lots of work. And my lab members, I had amazing lab members. Again, Professor William Foley, Kara, Karen, Jesse, and Pip, Hannah, Kim, Laura, and James. Thank you very much for friendly environment. I have amazing friends and colleagues. I met lots of people here and they were amazing. I learned lots of things about ecology because whenever I go to have a lunch with them, all they talk about is birds, reptiles, plants, insects, you know. having engineering background, you know, I learned lots of things and I spent quality time with them. I really appreciate it. Thank you guys. And last but not least, I thank to my parents, my father, my mom, my brothers, my wife and son and daughter who are supporting here. I'm sorry that I was late lots of times. Usually, like I was working like Sundays as well. Not Sunday, Saturdays. I was with them on Sunday, and I also thank all my relatives and friends back in Uzbekistan for their love, for their encouragement, and for their support. And I think I need to finish because my kids are getting bored already. This is the fusion of both datasets, by the way. So it's very good if you have this kind of three-dimensional model and you can play around and get information whatever you want. And it can be saved and it can be repeated. You can have like time series analysis and etc. Thank you very much for listening, everyone. Thank you. I appreciate it.
Fantastic, Shukar. Thank you.
Thanks. Thank you.
What we can do now is you can take questions in the room, but if you just repeat the question so that people listening on Zoom can hear. Otherwise, yeah. People on Zoom, if you would like to ask a question, just raise your hand and then I will turn on your microphone. Otherwise, you can type in your question and I can read it out for you. But maybe let's start, if you can, with a question in the room.
Is there any questions?
Great talk. That was really entertaining, especially on something that I know background. That's cool.
Thank you.
The two types of data, the TLS and ULS, are they not commonly together by the same people? So that was like a difficulty in putting it together. Is that right?
Yeah, exactly. I didn't see anyone who's used this data like TLS and ULS so far for ecology. So was that the...
Yeah. And so going forward, would you suggest that people collect both types of data? Or?
Yeah, that's a good question, actually. As you have seen, explained variance from both data is pretty much the same. Like they didn't improve the model dramatically. Therefore, and also ULS is much faster and much easier to collect. And we have lots of opportunities right now we have access to ULS data. So for ecology, I think ULS is enough. We don't need that much information, that detailed information as TLS captures, to my suggestion. So, yeah. Is there any other?
We can take one. Can we take one online? We've got Utkir Shemanov.
Utkir Shemanov. Okay. Hello, Utkir. Thank you.
Get my pronunciation.
It's my friend from.
Hello. Hello. Did you hear me?
Yes, yes, we can.
Hello.
Hi, Utkir. We can
I
hear
have a question.
Yes, please. Thank you.
So your research is very interesting, and I have one question about digital elevation model, and you identified that trees, yeah?
Yes, yes, yes.
Which fall down trees, yeah? How to identify it? You using special algorithms for these trees, or manually you find these trees?
No. I use two types of. One is it's LAS tools, we call it. It's a processing like software. It's like command lines and software. Another one is LIDAR, which is the R package, R programming package. So I use these two types of software to get this information, to extract information from this point cloud. Was that the answer?
How can filter? How can it's identify these trees? It's you can just choose like stones, also the different objects. How can identify especially its trees?
Yeah, you mean individual trees? I didn't identify individual trees because eucalypt has, very sparse point cloud, very sparse branches, and it's very difficult to delineate individual trees. Therefore, we just extracted from point cloud just information from the whole site, whole plot. But there are some if you are interested, I can give you some softwares which you can use to identify individual trees. It depends on the tree shape. If you have cone-shaped trees, you can identify it easily. If you have eucalypt trees, very sparse branches, it's very hard.
Okay, thank you. And did you compare also fusion TLS and ULS with time series, and how many times you spend for ULS and for TLS and fusion?
Yeah. For TLS, I spent 1 month to collect 96 hectare plots, like 1 hectare, 96 plots. And for ULS, it took about a week, basically.
Okay.
Thank you so much, Utke. I appreciate it.
Okay. You're welcome. Thank you.
Are there any questions? Yeah, Rocco has a question. Yeah, you can go. It's okay.
Thanks. I was wondering, whenever you could detect a bird species or a reptile species, you could also assign it to a species.
What's that?
Could you recognize, could you assign, whenever you saw a bird or a reptile with these techniques, could you assign it to a species?
I couldn't catch the.
Could you? Do you know what species was from the bird?
Yeah. We modeled individual species as well. They gave some information, some explained variants. I'm not sure if I understand.
I was wondering, how can you do that?
How can you do that? So you have.
I think it's not exactly with
No. It was just modeling. We have bird data from each plot, right? And we have a point cloud, which gives the information about the landscape. We just model predicted using those information, the bird abundance, basically. Because we have bird data already, bird abundance. Yeah, it's just a prediction model. Is there any? Yeah.
The last part you presented, it's a stupid question, but do you ever confuse kangaroos for trees or not? Like, I mean, because you had some pictures with kangaroos, and they are kind of the same shape as a fallen tree. And I was just wondering if your method is able to sometimes to confuse kangaroos and trees.
That's a good question. I tried a lot, like, to capture some kangaroos, but they never stopped, you know, like, it was so hard, but it would be great. One day, by the way, I was scanning and I went somewhere and saw that, like, lots of groups of kangaroos came and they were watching to my scanner. It cost $70,000. I was like, come on, don't touch that. Like, they didn't touch that, they ran away. But it's difficult to find kangaroos. Is there any other questions?
We do have one more online if you would like, maybe as a last one. Excuse my pronunciation, but Mamambek Raimov.
Yes, Mamambek. Hello.
Hello. Good morning, good afternoon. I don't know what time being. Thank you very much for the presentation.
Thank you, Mamambek.
It's very, very nice. I have only one question. It's I'd like to ask about a specific area, research area. You said that it's around 1 hectare land. Maybe I missed something. And I'd like to ask what's the specific area and why you chose only that focus target area. Is there any specific condition?
Yeah, of course.
Thank you.
That's a really good question. Actually, this Mulligan's Flat is critically endangered place, and therefore the government is like it's like a protected area. It's called sanctuary, and there are lots of bird and reptile introduction, and also to improve the ecosystem. You know, there are lots of treatments ongoing, like introduction of course DDB to make a habitat for reptiles, and also improving the ground vegetation. They excluded the feral, they excluded the grazers like kangaroos and etc. And also they built like fences to exclude feral animals like foxes, to protect these reptiles and birds from these feral animals. Therefore, it's very important if you have by the way, we have 1 hectare, 96 plots, which means 96, 1 hectare plots. Each of them is 1 hectare. So it's not only 1 hectare place. So if you have, and these plots are, lots of treatments are ongoing in this place, like as I said, and if you have data like this and you can predict those birds and reptiles, it's really important because it can improve like conservation. It can give some suggestions to future works for conservation and management things, the activities.
Okay, thank you very much. Wish you all the best and good luck for the steps.
Thanks, thank you very much.
Thanks, Mamambek. I think we probably have to end there for now. Shukrat, to let Carlos speak. So let me try and see if I get this right. So, Men Sizni Tabrikliman. Does that mean anything?
What is it? Sorry. I couldn't hear, sorry.
Men Sizni Tabrikliman.
Ah, Katta Rahmat. Katta Rahmat, Thomas Big. I appreciate it.
All right.
It means just congratulations, basically.
Fantastic.
Thank you so much, Michael.
And we'll have to move on now to Carlos, but it's very nice just while the transfer is occurring. So there's 54 people watching online, so it's a really great attendance. And I can see from guessing based on surnames, there's a healthy mix of people here for Shukrat and Carlos, and I hope everyone carries on watching.
Thank you so much. Thank you so much.
It's good to see that Carlos dressed up for the occasion.
Wearing the most important uniform in the world, isn't he?
Can you see that, Mike?
Yes, that's
okay.
All right. So the gag that I always do when I introduce exit seminars is that I like to dig up the first point of contact, the first email that I ever got from the student. And I like it because, of course, the student could never know, you know, if it pans out and they come to do a PhD here, they could never know that this would be divulged to the department. So I like that bit of it. So I'm just going to read you a little bit of this. Dear Dr. Keogh, my name's Carlos Joaquin Pavon Vasquez. I'm currently studying a master's degree at the National Autonomous University of Mexico. Then he goes on and tells me a bit about what he's doing. I'm interested in doing a PhD at ANU. Would there be any chance that you're my advisor? and I really liked that turn of phrase, and I thought, well, I don't know. And then he says, thank you for your attention. Have a nice day, Carlos. Now, normally I'd write back and say something along the lines of, no, there wouldn't really be much of a chance. And you know, we'd start a little dialogue maybe. Just, you know, I guess to put this in context, I made Damien do a master's first, just in case, just to check it out. I gave Ian a hard time as well. But I had a different interaction with Carlos, which is what I wanted to tell you. So I pretty quickly just wrote back and said, you have an excellent track record. I'm interested in lots of things, and what you have done and are doing and want to do all fall in the general area of things that we do in my lab. And I pretty much said, you can do whatever you want if you can get in. And this was about 7 years ago. We were in contact for a couple of years before Carlos actually came. And he did and does have an amazing track record. So Carlos has published something like about 15 papers, another 15 natural history notes. He's an amazing artist. He's a musician. He's an all-around nice guy as well. And he's done a really amazing PhD too, which is what he's going to talk about today. I'll just say something really brief, that Carlos has broad interests, and he mentioned this in his abstract, that he's interested in why do organisms look the way they do, why do they live where they do, and why are some groups more diverse than others. And he was able to touch on all of those things in his PhD using an iconic group of Australian and Old World animals, the famous goannas. So I'll hand over to Carlos, and also mention too that actually Carlos will be leaving us early next year. He's already scored a 2-year postdoc in the US, looking at the genomics of some amazing American lizards as well. Over to you.
Thank you for the very nice introduction, Scott. I feel a little bad about the photo I'm using of you in my acknowledgements. I was expecting you to be harder on me. So. Well, thank you all for coming, and those on Zoom, I'm very happy to be here, even though my stomach disagrees. This morning it was misbehaving a little bit. But anyway, let's get into it. So as many people in this room, my interest in nature comes from a very young age. I used to go on hikes with my family. That's me. And then of course I went through a dinosaur phase, but I think I never really grew out from it. Then I always loved animals. This is my goat, Clementina, which I used to keep in my house in the middle of Mexico City. My parents, of course, were not impressed. And I always liked reptiles, but with time, this little taste became a little more refined. And I ended up doing some taxonomic work back in Mexico, starting from around 10 years ago. So as Scott mentioned, the questions that I'm interested in are pretty basic. I'm interested in why animals look the way they do, why they live in the places where they do, and why some groups are more diverse than others. And here's the mammalian phylogeny, which provides a great example. We have the monotrends, which are the echidna and the platypus, which there are only around 4 or 5 species. Then we have marsupials that are a bit more diverse, and then we have the placentas that just went crazy, and we have things like giraffes and whales and bats and humans. And we can look at these questions traditionally from 2 different angles at 2 different scales. For example, if our question is why animals look the way they do, we can look at a more ecological microevolutionary scale. Here are two species of beetle that have two morphs. And we can see that when they live separately from each other, they look very similar. And when they live together, actually one of them becomes larger and the other one becomes smaller. And this is probably due to character displacement and competition. Or we can look at a broader scale, like here is the adaptive radiation of cyclid fish from Lake Tanganyika. And we can look at all these different adaptations and how they relate to what they do in their environments. And recently, there's been people trying to link these different scales and different approaches to understanding nature. And this is an example that I like from last year, in which they look at the species diversity in two families of shark. And they found that when the number of species in one of the families went up, it went down on the other group. And they think this is because of ecological interactions and competition. Another excellent example is this paper by Ian and collaborators, in which they show that models of morphological evolution, in which they incorporate competition, are better suited for explain better morphological variation in monitors and in marsupial carnivores. So now I'm going to present to you the best organisms on Earth, the monitor lizards. There are around 80 species of monitor, and they occur in Africa, Asia, Australia, and numerous Pacific islands. And the body plan is relatively conserved. They all have 4 well-developed limbs with 5 fingers, relatively long tails. And this is a very dramatic example of this. These two used to be subspecies of the same species until 1994. And now we know that actually they are separated from each other by 30 million years of evolution. Well, even when monitors have a very conserved body plan, there's great variation in one key aspect, which is body size. Here is the smallest monitor, which is the Dampier Peninsula in Goana, which is around 20 centimeters long. Then we have the largest living monitor, which is the Komodo dragon, around 3 meters in length. And then we have the huge Megalania, which is an extinct monitor that was around 5-6 meters in length. Now, if we compare this variation in body size with the variation in primates, you will see how fast this body size difference evolved in monitors. Here we have a mouse lemur and we have a human, and this size difference took 75 million years to evolve, but in monitors, this huge difference only took 20 million years only. And also within their body plan, there's some variation in the relative proportions of the body. So we have some species with very long necks and others that have a shorter neck, and some that have very long tails like these 3, and others have shorter tails like this fat guy over here. And some of this variation is related with how they interact with their environments. On the left, we have 2 species that have very long tails and long limbs, and they live either on the canopy, like this one over here, or they live in rocky escarpments. Then we have some semi-aquatic monitors that have the nostrils on the top of their heads so that they can breathe when they are underwater. We have this tiny, tiny monitor that has a prehensile tail, which it uses to grab onto clumps of spinifex. We have this very fat terrestrial monitor. And we have this other species that uses its spiny tail to lodge into rock crevices. There's also some variation in diet. Most of the monitor lizards are predators, and they are either insectivorous or feed on vertebrates, like this Komodo dragon, which is feeding on a buffalo. But there are also some monitors that eat fruits. And when I was looking for photos of these monitors, this was the first result in Google. I don't know why. and then the second result was this one, which is not much better, but anyway. So this is the outline of my thesis. I have 5 chapters. The first one is about the diversification of varanids, the whole family in space and time. This one is hopefully about to be submitted to evolution. And I have another chapter on the evolution of ontogeny. Another chapter looking at hybridization in a group within the monitors. And these 2 I talked a little bit about before in front of you guys. So I think I will be more, put a little bit of less time on those. And then we have these 2 chapters, which are focused on the phylogeography and taxonomy of 2 species complexes. And they are very similar to each other, so I will only present one of them. So let's go to the first chapter. So we build a phylogeny of the monitor lizards. And the first thing that comes to mind is that there is great variation in richness between each of the clades within the monitor. And the three more diverse clades are this one, the one from Australia, the Australian clade, the one from Papua New Guinea and surrounding islands, Papuan clade, and one from the Philippines and adjacent islands, which I've called the Malaysian clade. And all of these occur in Australasia. When I refer to Australasia, I mean Australia, the Philippines, Papua, and surrounding islands. So trying to look into what's driving these differences in richness, we fitted several diversification models and then averaged them to get an idea of the diversification rates and compare them between the monitors that occur in Australasia and those that do not. And we found that the speciation rates are much higher, well, not much higher, but they are higher in the Australasian monitors. But then when we take a closer look at each of the clades in Australasia, we see that there are some differences in the speciation rates. We see that they are especially high in the Papuan and Malaysian clade, while those of the Australian clade are much close to the background speciation rates. The background speciation rates for the family are this black one on the top. Then here we are looking at morphological data. These are 4 data sets on body size, body shape, head shape in dorsal view, and head shape in lateral view. All these data set was like 2 megas on like Shokirov's terabytes of data. And then we can see that Australasian monitors are more morphologically diverse. They are represented by these orange hotspot and these orange polygons compared to the non-Australasian monitors, which are these green polygons and this green hotspot over here. But then again, we can look more closely at each of these clades and we see that most of the morphological variation is found within the Australian clade, which is this pink one over here and over here. And when we compare the rates of morphological evolution, we found a surprise because we see that they're actually much higher for the Papuan and Malaysian clade. So diversity is accumulating faster than in the Australian clade, in which the rates are again closer to the background rates for the whole family. So monitors appear to be more diverse, both in number of species and in morphology in Australasia. But it seems like each of these Australasian clades has diversified in a particular way. So first we are going to look at the Malaysian clade, which is mainly found in the Philippines. And we found out that the rates of speciation are negatively correlated with time and the number of species. So as the diversity in this group increases, species are forming less fast, they're forming slower, probably because of increased competition. And then when we look at the rates of morphological evolution, we see that there's no relationship at all with time and the number of species, so they are pretty constant overall. And then we use ABC and linear discriminant analysis. ABC stands for approximate Bayesian computation, to see what has been the geographic speciation mode in each of these clades. And we found that for the clade, Malaysian clade, it has been mostly allopatric vicariant, which makes sense when we look at the geological history of the Philippines. So it has been proposed that during the Pleistocene, there were sea level fluctuations that sometimes connected and disconnected these islands in the Philippines. So in summary, it seems like this radiation has been fast, and I think this has to do with the fact that these monitors live in islands, which are very unstable environments. But this apparently has been non-adaptive. It has been mainly driven by vicariant speciation, and there has been very limited ecomorphological differentiation. Most of these monitors are large and semi-aquatic. Then we are going to look at the Papuan clade. And we can see that the rates of speciation are positively correlated with time and the number of species. So these guys are still going strong, still diversifying very fast. And the rates of morphological evolution, depending on the character that we look at, they are either positively or negatively correlated with diversity and time. And when we infer the geographic speciation mode, we found that founder event speciation has been the main mode of speciation here, which makes sense because if you look at the distribution of these monitors, a lot of them are endemic to little islands in the middle of the Pacific. So they could not have arrived there if it wasn't for water dispersal. So in summary, it seems like this radiation has been, again, fast. Remember that the rates of speciation and morphological evolution are higher than background rates. But it's a young radiation that apparently hasn't reached an equilibrium state, so I think it will be a good model to study the early stages of radiation. Finally, we are looking at the Australian clade, and we can see that the rates of speciation have decreased with time and the number of species, which is consistent with fast speciation following ecological opportunity, and then declining as niches become filled and competition is more intense. And same for the rates of morphological evolution, they decrease with time and the number of species. And dispersal was, founder event speciation was inferred to have been the main mode of speciation here as well. And these are the results of an analysis that is trying to look for adaptive optima in the monitor lizard radiation. And in other words, these different colors are telling you how ecomorphologically diverse monitors are. And we can see that there are more adaptive shifts in the Australian radiation compared to other clades. So in summary, it seems like the Australian radiation has been slow, and I think this has to do with the fact that it has been mainly based in Australia, which is one of the most stable continents on Earth. It's sitting in the middle of a tectonic plate, and there's not much orogeny or anything going on really. But it seems to be adaptive, and I think that the ecological opportunity may have arisen through a combination of the environmental heterogeneity that is present in Australia, coupled with reduced competition and predation from diurnal mammalian predators, which are more and more diverse in Africa and in mainland Asia than they are in Australia. And so when we look at the Papuan and Australian radiations more closely, there's something very funny going on. We know that Australia and Papua New Guinea have been intermittently connected for the last million years. And despite that, we see very little immigration from the species occurring in Papua to Australia and vice versa. And the few species from one clade that have invaded the range of the other clade only make it to fringe habitat, living in mangroves or strips of coastal rainforest. So we think this may have something to do with the fact that these radiations have already diversified and occupied ecological niche space in these two respective islands. And so the few species that can make it to the other place are failing to diversify. Then we fitted some evolutionary models, and we indeed found out that models in which the immigrants have no speciated at all were preferred. Now we are looking at morphology. Here I'm presenting body size, just to give you an idea of what's going on. The immigrants are represented by these triangles, and this is the variation in the monitors which they coexist in their secondary range. And we can see that the invading species are in general different from the species that already live there. So we thought that this could have something to do with biotic filtering. So only monitors that were doing things a little bit differently could colonize these areas that already had monitors and survive there. But actually, when we tested these differences statistically, we found that the monitors that occur from one clade, coming from one clade and the other, are more similar to each other than expected given the baseline differences between these two clades. So we think that it may be instead the environment that is deciding what characteristics the monitors that successfully invade have to have. So in summary, for this chapter, we found out that geography and ecological opportunity have apparently shaped the diversity of Varanidae, that ecological interactions matter for microevolution. They can have long-term evolutionary consequences, that in situ diversification of one lineage may prevent the further diversification of immigrant lineages, and that environmental conditions may be dictating which species can immigrate to new habitats. Now on to my second chapter, which is about the evolution of ontogeny. So we have all this diversity in body size, right? And we have also this diversity in body shape in monitor lizards, and we may be asking what mechanism is promoting this differentiation. So basically, this chapter is about comparing how different species change in shape as they grow. We know that for most species, when they grow, they have very different proportions. So adults of humans have smaller heads and they have longer legs compared to babies. But there are some other species in which the adults pretty much look like big babies. So we are going to be comparing these patterns between the different species. So this is just a PCA, and the average shape for the juveniles of each species is shown by the white dots. And then the average adult shape is shown by the black dots, and there's a line connecting them. And you can see that in most cases, monitors are, as they grow, they get shorter tails and shorter heads. And for the limbs, they also get shorter and thicker. But there's one clade that appears to be doing things differently, and it's this one shown here in green, which is going in the opposite direction. Same for the limbs. So these monitors, as they grow, they are becoming more gracile, more thinner. And this is a group of specialized, strictly arboreal monitors. And then we look at what ontogenetic changes are underlying the differentiation at different evolutionary scales. And we found out that differentiation, morphological differentiation between clades is mainly caused by changes in the direction of ontogenetic change. By within clade differentiation is mainly caused by changes along the same trajectory. Some are cutting it short, so they look more UV-like, more juvenile-like, and others are extending it, so they look more adult-like. And finally, this is a PCA showing the main characteristics of the ontogenetic trajectories. And we can see that most of the variation is represented by this clade, which is the one of the Australian clades. So in summary, it seems like ontogenetic shifts are important for ecological specialization to occur, like we saw in this very specialized arboreal monitor. That morphological differentiation at different scales is caused by different ontogenetic shifts, and that ontogenetic evolutionary liability may be underlying adaptive radiation, like in the case of the Australian monitors. So the first chapter is about hybridization in a group of monitors that includes the Komodo dragon and its close relatives. So there have been people doing the phylogenetics of these monitors throughout the years, and depending on the kind of data that they use, they get different relationships. And usually this is a sign that there's something funny going on, and in a lot of cases it's because of past hybridization, and so gene flow between two different species. And then we performed several analyses, and we actually found out that the sun monitors, which are these 4 species over here, have a proportion of the genome around 7% coming from the Komodo dragon. And then looking at the morphology, that's the morphology of sun monitors. And we can see that it's intermediate between their closest relatives, which are these over here, and the Komodo dragon over here. So this could be telling us that past hybridization resulted in morphological intermediacy. The hybrids are intermediate between both of the parents. And then we tested this with a model fitting approach as well, in which we tested models in which hybridization leads to morphological intermediacy, which are these orange ones over here. Then other models where hybridization results in extreme phenotypes in the hybrids, which are these bluish models over here, and then the boring tree-like models in gray over here. And we found that for most of the characters that we recorded, the preferred model was one where hybridization resulted in morphological intermediacy. But you may be asking, those who know about monitors, if the Komodo dragon lives here in some remote islands in Indonesia, and the sun monitors live here in Australia, then how did hybridization occur? But these stars are some fossil records which some people have assigned to the Komodo dragon. And it's cool to see now that with independent molecular data that the Komodo dragon may have lived in Australia because they will have to be in contact with the sun monitors to have gene flow with them. And we propose that the Komodo dragon originated in Australia, then migrated into these islands in Indonesia, and then got extinct in Australia. So in summary, it seems like hybridization can have long-lasting effects in the phenotype, even when it's followed by speciation, like in the case of sun monitors. The Komodo dragon hybridized with their ancestor, but you can still tell, and then this ancestor is speciated into different species, but you can still tell that their morphology has signs of this hybridization event. And we also show that the detection of hybridization can illuminate biogeographic history. So finally, I'm going to be talking about the phylogeography and taxonomy of these monitors, the spiny tail monitors. And then Varanus acanthurus, the spiny tail monitor, is composed by 3 subspecies. We have Varanus acanthurus from the monsoonal tropics. Then we have Varanus acanthurus brachyurus, which is widespread in the arid zone, and Varanus acanthurus insulanicus, which is found in these islands in the top end. And we use thousands of nuclear molecular markers to look at population structure in this monitor. And we found out that there's 6 populations, which are shown here with different colors. And we also found out that the Varanus acanthurus is paraphyletic with respect to Varanus barriggia, which is shown here in blue. This is a principal coordinate analysis of the nuclear data. And we found that all this structure is found in the islands in the Kimberley. So these populations in the Kimberley are very different from each other. Beck, I think, would be happy to hear that. But the thing is that these islands only became islands very recently. But it seems like they have harbored biodiversity for a long time, and there are endemic lineages occurring there. And then we perform a fits difference analysis, which is trying to see if there are significant fits differences in the genomes of these animals. And we found out that there are no differences between these 2 populations, so they can be collapsed into Varanus acanthurus. Then all these 3 populations can be collapsed into Acanthurus brachyurus. Then we found no significant fits differences between Barriggia and Acanthurus insulanicus, and this population from Cape Crawford, which appears to be isolated from the others. So it appears like the taxonomy of Varanus acanthurus will end up looking like this. We have Varanus acanthurus occurring in the monsoonal tropics. Then we have Acanthurus brachyurus, which is widespread in the arid zone, and they appear to be in contact over here. And there appears to be significant gene flow going on there. So probably they should just be kept as subspecies. Then we have Barriggia and Acanthurus insulanicus, which probably should be synonymized. And we have this population from Cape Crawford that probably represents an undescribed species. So in summary, there is unrecognized species level diversity in the Varanus acanthurus complex. And we show that it's important to sample contact zones so that we can detect any potential gene flow between the populations that we are studying. And that the Kimberley Islands, while they only recently became isolated, they have been important refuges for a long time. So that's it. I think I went too fast as usual. Well, I want to start by thanking Scott.
I look exactly the same.
So I think that you only accepted Damien in the lab because he also had a ponytail. Well, I want to thank Scott. He has been an amazing supervisor. The years here have been awesome. And Scott has always been there supporting me, like both personally and academically. And I couldn't think of having a better supervisor than him. I also want to thank the Kiosk lovers. I want to thank Damien Escarré, who once looked cute, believe it or not. I also want to thank Ian Brennan. I thank Connie, the honorary member of the Kiosk Lab. And I thank Potter, who is planning to kill someone because they cooked with basmati rice instead of jasmine rice. And I wanted to look for another photo, but he was like, oh, use this one. Of course, the one where he looks badass, you know. And I also want to thank Bank. The Bank, no? Who's not here. So. And Mitzi. I want to thank my ETIT seminar brother, Shokirov, for agreeing on presenting with me and see all this ridiculous bullshit. I also want to thank my supervisor, Tano, Craig, and Rob, those who took a bureaucratic bullet for RSV, Scott, Mike, Adrian Nicotra, Rob, and Celeste, and the E&E Dream Team, Audra Johnson, Jack Egerton, and Red Skid, IT, cleaning and building maintenance, for keeping all running from behind the scenes. The mastermind behind my PhD, Maria Angel, who she actually helped me with analysis and stuff. She recorded some of the morphological data and digitized specimens. So thank you. I want to thank my co-authors. A lot of the work that I presented here was made by them. So I thank Ian, Alex, Daniel Ferrer, Steve Romevan, and Paul Dutty. The Moritz and Cardillo labs, they have been very supportive throughout the years and provided very useful discussion. Staff from all these different collections, Stephen, Susaya, Damian, and Octavio for allowing me to use their excellent photos. And for funding, I want to thank ANU, the government, and the ACTR Petroleum Association. Well, I want to thank you all for being here. It's been a fun time. And it will be very hard living to the US. I speak to a lot of you and told you that I don't really want to leave. There's nothing wrong with New York, Ian, but still I've had a great time here. I also want to thank my friends back in the Western Hemisphere, my friends and mentors. Especially I want to thank Adrián Nieto, Uri Garcia, Luis Canseco, Levi Gray, and Britt White, because they supported me when I was no one. I'm still no one, but, but when I was just a second semester undergrad student. I want to thank my family. It's been great to be a part of this very big and supportive family, especially my core family. I want to thank my dad, my mom, my brother, Maria Angel, not yet. But I will switch to Spanish. Bueno, quiero agradecer a mi familia, sé que están viéndome por acá, porque sé que hicieron muchos sacrificios para que yo pudiera estar aquí, y este logro creo que es más suyo que mío. También quiero agradecer a María Ángel, porque me ha apoyado mucho durante todos estos años, soportó todas mis locuras, y ahorita ya podría decirse que es una herpetóloga honoraria. Y quiero dedicar esta presentación a los que ya no están con nosotros, que desafortunadamente se fueron cuando estaba, mientras yo estaba aquí, y que siempre los voy a querer mucho. And thank you. Are there any questions?
Qué padre, Carlos.
Size where I would expect there to be persistent shared segregating alleles between even really highly divergent images. So is that a thing that you do commonly?
Well, yeah, you expect that with gene flow there will be allele sharing, you know, and it only takes a few immigrants to homogenize populations. So yeah, the rationale is that fits differences are an indication of lack of gene flow. And to see if they are significant, you compare them against simulated values. But there's still a lot more that I had to do to say, oh, these are different species. So I have to look at the morphology. I've already done some analysis, and it appears like yeah, there are consistent morphological differences. And you can also more specifically model gene flow to see if there's any. Oh, sorry, I forgot to repeat the question for the people in Zoom, but he was asking what's the rationale behind using Fitch differences to distinguish between populations and propose that they are candidate species. Any other question? Yeah.
Great.
Thank you.
In your chapter about the differences between the babies and the adults,
Mm-hmm.
Why do the babies look so different to some?
Oh, yeah, that's a good question. So Lauren is asking why the babies of the monitors look so different from the adults. And the answer is that it appears to be related with ecology. So most of the juveniles are arboreal because it's more safe up there in the trees, you know, from predators, including other monitor lizards. So if they are doing things similarly, you expect them to be more similar between the different species at that age. And actually, that's what we found. I didn't show it here, but we found that the juveniles of different species are more similar to each other than are the adults. And then when they are adults, they come down from the trees, but some stay there, and others come down from the trees and start doing other things like some semi-aquatic species, terrestrial species. Something?
I have a question about the hybridization of just having Komodos in Australia. It's crazy and amazing. Can you do ancestral reconstruction to reconstruct into what the ancestors look like in Australia? And I don't know much about hybridization. Are there particular phenotypic traits that resemble, that you see in pangolins that resemble?
Yeah. Well, for that chapter, Fonte is asking if we can estimate how the ancestral Komodo dragons looked when they were living in Australia, and also what was the second part? Yeah.
What would be like the similarity?
Yeah, and if there are any similarities between the sun monitors and the Komodo dragon, given that apparently there's been some gene flow. So for the first one, I think it's challenging to model a genetic change, so change along the same branch, because you can do it for phylogeny because you have two sources of information, so you can tell but I think that if you wanted to see how Komodo dragons looked when they were in Australia, you will need to look at the fossils. And these guys that did the fossil work identifying that there were Komodos in Australia say that body size at least has remained pretty stable, which is surprising because people before thought that the Komodo dragon was a case of insular giantism, but it appears like they've always been large. And the second question, we performed some convergence analysis between the Komodo dragon and the sun monitors, which will tell us if they are more similar than expected by chance, and that could be due to convergence or hybridization. And if I remember correctly, most of the characters were more similar to the Komodo dragon than you would expect by chance. And the characters that I measured were like tail length and head length, limb length. Yeah.
Thank you.
Yeah, no worries. Any other question?
Can you hear me, Carlos?
Yeah.
Can you hear me?
Wait. Now?
Can you hear me?
Yes.
Okay. I just quickly wanted to say to people online, if they would like to ask a question, if they just raise their hand. And if anyone wants to ask a question in Spanish, I'm sure you can translate. So, si quieres hacer una pregunta en español, no hay problemas. Carlos, él puede traducir.
Uri, Adrián. No, they are shy.
Oh, Miguel. Okay, Miguel Herrera.
Oh.
Hey, can you hear me? Well?
Simón.
Yeah, okay, perfecto. So maybe this is out of the scope because you were like analyzing changes in or similarities in the genes, right? However, do you think that the monitor lizards are going to be able to cope with the fast changes in the environment? Meaning that I know that this is a complex system, so evolution takes a bunch of time. However, based on the changes that we are seeing in the environment and do you think monitor lizards are going to be able to adapt to this? Or what do you think is going to be the future of the monitor lizards?
Thank you for your question, Miguel. This is my cousin from Mexico.
Yeah.
Well, I think that monitors have shown to be very adaptable. So I think there's a good chance that they will survive unless, there's a giant asteroid coming and killing everyone. But I think that with climate change and everything, there's a good chance that some of the species are going to go extinct. There's been a recent paper on the Komodo dragon that showed that they are actually very vulnerable to environmental change. So they may actually be one of the first species to go out. Also, they have a very small range, so they are very vulnerable. And for some of the monitors in Australia, an additional threat is the presence of cane toads because they are already here. and which are some poisonous toads that apparently can, if you eat them, you can get killed, and some monitor populations appear to have gone down since the cane toads arrived in Australia. But yeah, to answer your question, I think that some species are going to be more prone to extinction with environmental change, but others may well survive for a little bit at least. Yes. Thank you.
Okay, thanks.
Thanks, Miguel.
Yeah.
Ian?
Spends a lot of time looking at monitors. What's your favorite one?
I think that's a really hard question. Ian is asking what is my favorite monitor lizard species. It's hard. I really like the really small ones, like the Brevicada, and I also like the arboreal ones, the Prasinus group, because they have all these different adaptations. One thing that I didn't tell is that they also have a sticky black substance on their hands, so they actually can stick to the trees. And I also like the big ones. I like them all. But I think it will be between the three of those, like the really small ones, the really big ones, including the crocodile monitor from Papua New Guinea, because it's really big but little known and arboreal, and the Prasinus group. Yeah.
Okay, there's another question here from Levi Gray.
Hey.
Hey, how's it going, Carlos?
Not bad, not bad. How are you?
Pretty good. Congratulations. This was awesome. You kind of mocked anoles earlier, calling monitors the best lizards. But I just wanted to ask a question about, like, so one of the things we see in anoles are like increased speciation rates where there's specialized lineages. So like oddball lineages tend to get a lot of like, you know, small range endemics.
Mm-hmm.
And like, do you see that, or are there any signals in the morphological data where like specialists tend to diversify faster?
Yeah, I think there are a couple of examples there. One is the tree monitors. So in the Prasinus group, actually there's like 6-7 species, and there may be a little more. And yeah, I think it's because they are strictly arboreal and very bad at moving on the ground. So you will expect that there will be, that will make speciation easier. And the other one is actually the monitors that live in rock crevices, like the Acanthurus that I just showed, because also they are very fond of their little rock habitats, and they don't go out in the open as much. So, you know, that promotes speciation as well, as we see in Cenosaurus back in Mexico.
Right.
Yes, thanks, man.
No, good answer. Thank you.
It's a bright answer.
Are you already drunk?
Well, you know.
Yeah, I know. Yeah. Don't embarrass yourself anymore. Just mute yourself. Thanks, man. Any other questions?
So, well, I think it's very interesting that some monitor lizards are known, quite recently, that they have toxins in their saliva that has strong implications in their way of foraging and all that. And I mean, this is off topic of your thesis, but you know quite a bit about the natural history of monitors. So how widespread is this? Is there mass variation, or there's a lot to discover about this still?
Yeah, I think there's a lot that needs to be tested formally. Like, there's even some people that say like, oh, every organism has some things in their saliva, like even humans, that start predigesting food. But yeah, there's been some analysis that seem to show that actually the venom can have a high impact in coagulation. And I think you're right. I think it could have very important ecological and evolutionary implications. So maybe the monitors that are like seed and weight, like well, it's very far away, but the fat monitor that you show me the photo of, and the Komodo dragon, maybe they are seed and weight predators because they rely on the venom. And it has been shown, I think, the presence of venom has been tested on the Varanus clade, which is Komodo dragon and close relatives, in Odatria, which are the dwarf monitors from Australia, which are also closely related to the Komodo dragon. And I think there's some study on Varanus bengalensis, I think, which is from another clade from mainland Asia. So it appears to be pretty widespread. And if you believe Brian Fry, then maybe being venomous will be the ancestral condition, and instead they will lose it sometimes. Yeah. That's it. Thank you.
Thanks, Carlos. I think I'll just end the online meeting now. So thanks to everyone for coming along, and I hope you have a good party there.
A good what? Party?
A good party.
Oh yeah. First we have to play soccer, but yeah, after that, yeah.
And I think I should make special mention of your tia Ana, who was sending quite a few messages through for you in support.
Oh, thank you. No, gracias, tia. Thank you.
I think she's very proud. So have a wonderful time. Congratulations to both of you.
I hope you were here. Thank you.
All right.